All right, thank you everyone for joining. I'm going to go ahead and get started. I'd like to welcome you to today's webinar on assessment and control of exposure to reactive chemicals in construction. This will most likely be part of a series over the next year or so. So we are calling today part 1, and it will be focused on spray polyurethane foam applications. So my name is Jessica Bunting, and I'm the host and tech support for this event. Before we get started, I'm just going to go over a couple things. First, if you are having any technical difficulties with the phone or web piece, please contact me using your chat box or by responding to any of the WebEx emails you've received. I think WebEx should have just sent a reminder email about a half an hour before the event started, so you can just reply to that. If you're having trouble hearing through your computer speakers or headphones, I recommend calling in using your phone instead, and that call-in information can be found on your Quick Start tab or Event Info tab or in any of the emails that you've received. We will be taking questions at the end of today's presentation. You can enter them at any time in your Q&A or chat box, and we will do our best to answer as many as possible at the end. I will be reading these out loud, so please make sure that your questions are addressed to everyone or to me, the host, and not privately to our speaker. Finally, today's webinar will be recorded, so if you're interested in reviewing or sharing the information with colleagues after the event, we will make that link available to everyone via email. Our speaker today is Dr. Dhimiter Bello. Dr. Bello is an associate professor of exposure biology in the Department of Public Health at UMass Lowell. He is also a visiting scientist at the Harvard School of Public Health in the Center for Nanotechnology and Nanotoxicology. Dr. Bello's research and training are interdisciplinary and draw heavily from organic and analytical chemistry, as well as exposure sciences and toxicology. His current areas of research include reactive chemicals in construction, the toxicology of copier and printer-emitted nanoparticles, oxidative stress, and biomarker discovery and application utilizing non-invasive biological media, such as urine and sweat, extracellular vesicles and miRNA, and specific biomarkers for isocyanates. And with that, I will hand it over to you, Dr. Bello. I'm making you the presenter right now.
Thank you, Jessica. I would like to thank the participants for making time out of their busy schedules to attend the webinar. I would also like to thank the CBWR for giving us the opportunity to share our research findings with a broader community of researchers and other stakeholders interested in this line of research. I would like to mention briefly that because our research hasn't been published yet, we ask that you do not publicly share the data at this time. So I will spend most of my time discussing the results and only a small amount of time providing details about the methodology, only to the extent that we need them to understand the results. In the interest of time, I will try to be brief. Hopefully the Q&A section at the end will help me address some of those issues. This project focuses on isocyanates and epoxy-based systems in several construction trades, such as SPFs, industrial coatings, and other systems, including the new technologies. And the main focus of the project is really to identify and promote better work practices and controls so that construction workers and crews can do their job safer. The reason why we are looking at these systems, in particular isocyanates in SPF, is largely because of the well-understood health effects of isocyanates, which especially affects respiratory sensitization and asthma, but also other respiratory disorders, skin allergies, skin irritation and burns, and contact dermatitis. There has been a growing understanding of the role of skin in systemic sensitization, in addition to skin localized effects. For that reason, we are interested in understanding both inhalation exposure and skin exposure to these workers. SPF usually is applied as a Part A and a Part B. I'll provide more details as background a little bit later, but in the context of health concerns, Part B is a mixture of different ingredients, which includes amine catalysts, flame retardants, and blowing agents and surfactants, a number of proprietary additives, as well as may include reaction by products, and aldehydes have been mentioned as being one of those byproducts. We have a more limited understanding of the potential health effects resulting from exposures to Part B components. Of that mixture, the amine catalyst and the flame retardants have become sort of chemicals of interest. Amine catalyst because at least some of the ones we know are used in SPF applications have toxicity profiles that include irritations and the halo vision effect. Possibly one of them may be a sensitizer, as well as systemic organ toxicity such as liver toxicity and the like. Flame retardants, one of them, the so-called TCPP, included in SPF applications, is becoming of the chemical of interest because of some animal toxicity data suggesting potential involvement in endocrine disruption. Again, I would like to emphasize that our understanding of exposures to these chemicals from Part B in this cohort of workers and the health effects are not well documented or understood. As a background for SPF applications, a typical crew has a truck and two people, a sprayer and a helper. The isocyanate and the Part B are contained in separate drums, and they are pumped actively through separate hoses, sometimes several hundred feet through heated lines, and they are mixed at the point of the gun. And as a result of that mixture, the foam is sprayed as these aerosol droplets, which then start to react quickly to form cured foam. The typical tasks of this crew are site preparation and then spraying and then shaving the foam against the studs, sometimes, not always, and the last step is the cleanup. But there's a designated sprayer and sometimes a designated helper. The pictures on this slide show a typical set of SPF application in a construction crew, as well as the one at the bottom, a small spray job in a residential home and some remediation work. I'm mentioning this last one, essentially removal of a closed cell foam because the owner had complaints, as something that has come up repeatedly as a request for us to look at in terms of exposures and potential involvement. So we have conducted a pilot study on trimming of closed cell foams. And I want to mention that we have some data on that, but I don't think I'll be able to present those today. But hopefully we can address some of those in Q&A sections. We have worked with two sort of groups of SPF applicators. One group has been large contractors in new constructions. These have been homes or large buildings where construction is new. There are open spaces, and the crew has flexibility in access and in arranging to do this work. To the left, one can see the sprayer spraying the foam, and to the right, the helper shaving the foam against the studs. These large construction crews have dedicated safety professionals on site who oversee and assist with both preparation and safe application of such foams. The second category of SPF contractors has been small contractors who, in our experience, often engage in insulating, retrofitting, insulating old homes, such as in attics and other crawl spaces, as well as the bottom of the floor of the house. Typical for these applications is that space is very tight. These are crawl spaces. As you could see here, one of the sprayer is crawling on his back or sometimes on his belly to spray the foam. In these cases, the foam is not shaved or trimmed. But these smaller crews oftentimes have immigrant workers who don't speak much English, and they oftentimes do not have dedicated health and safety specialists to oversee these applications. For the reasons that I mentioned, our interest was to take a comprehensive look at the spray foam applications, and so our methods include monitoring of inhalation exposure, dermal exposure, urinary urine collection for measurement of urinary biomarkers, in addition to a comprehensive sort of data gathering of contextual information such as the activities and size of crews, duration of tasks, and even environmental conditions such as temperature, relative humidity, was it sunny or rainy. Before I mention the selection of different techniques we use for collection of inhalation and dermal exposure, and for urine for that matter, I would like to mention an important consideration in sampling for reactive chemical systems, of which isocyanates are one of those. Because these are highly reactive systems, as soon as the isocyanate is mixed with the polyol, it starts to react chemically and forms these intermediate prepolymeric species, which over time then become newer species. In this process, the isocyanate group is consumed, and the new species that are formed may have additional isocyanate groups, but we are ending up with the formation of new species. And the gold standard for effective collection of isocyanates has been a liquid sampling system that has a derivatizing reagent which reacts to the isocyanate and stabilizes it so that then the isocyanate can be measured at a later time. Impinger has been, with a backup filter, has been sort of the method of choice in these systems, but impingers are not suitable for personal sampling and they are cumbersome. Fortunately, at the time of the study, a new sampler called SIP10 was introduced in the market and evaluated and found to be suitable for sampling SPF applications, and we adopted the SIP10 sampler as the default sampler for personal sampling of isocyanates. We are perhaps the first group to adopt it in large scale besides the group in Montreal. The SIP10 collects on liquid, and it's personal and well tolerated by the workers. In addition, for dermal exposure, the same logic of needing to have a derivatizing reagent on a collection medium was considered important. We developed in prior work cotton glove impregnated with our derivatizing reagent called MAP, which is recommended by NIOSH methods. And these glove dosimeters were worn on above or on top of the regular gloves that typically nitrile that the workers used, and collected after a certain period of time and placed in a jar with an extraction solution. And an example of some of these jars is shown below. An example of this glove dosimeter at the end of sampling is shown in this little box to the right. We also collected some area samples, actually side by side of impinger samples that contain the MAP reagent and a novel chemistry that we have developed in our lab in collaboration with Dr. Stryker at NIOSH called the DAN method. And the idea of collecting samples, area samples, was to judge whether bystanders would be exposed. while entering this active SPF zone. We collected urine pre-shift, usually at the beginning in the morning, as well as at the end of the shift, and urine was collected to measure biomarkers of isocyanates and to judge whether exposures and uptake into the body had occurred. This figure shows a sprayer wearing the gloves as well as the SIP-10 sampler during a pause when they stop and try to clean the spray gun, an activity that happens frequently, during which there is some potential for contact with raw materials such as the isocyanate. We collected information about the safety data sheets of materials that were used in our sites, and one could see in this table that for the most part, Part A is a polymeric MDI material that also contains the isomers of the monomeric MDI. There is no variation in terms of the type of isocyanate used in Part A, and this is important. However, composition of Part B is a lot more variable, and it contains, in addition to blowing agents and surfactants and the polyols, other proprietary or trade secret ingredients, and a flame retardant, TCPP being a common one that surfaced in these cases. So based on that, we set up analytical methods in our lab to measure important or relevant species related to MDI. All of them are done using ultra-high performance liquid chromatography with tandem mass spectrometry in electrospray ionization mode for the interested chemists in the audience. So we monitored for transitions of phenyl isocyanate, the 3 isomers of MDI, as well as the main, one of the main trimers, the one that we see frequently both in the bulks and in spray foam insulations. We also have an online UV detector prior to the mass spec, for which we collect both UV spectra of the different peaks and monitor absorbance. And the UV detector can be used to calculate total isocyanate content that includes not only these measured peaks, but also other new species that are formed. Isocyanate biomarkers were measured using, again, liquid chromatography mass spectrometry. We have adopted a commonly used method, the only one useful at the moment, which is hydrolysis of urine under acidic conditions, and then quantification of the diamine corresponding to the diisocyanate. There is a derivatization step in between that makes quantitation easier. This illustrates a panel of the markers we measure routinely in all the samples. In the context of SPF applications, it is the 4,4' MDA and the internal standard that are relevant. So based on our field observations, we recruited 53 participants, of whom 40 were sprayers and 9 helpers. We also got 4 bystanders to participate. About half, a little bit more than half of these participants wore supplied air respirator, and essentially all of them were related to large contractors who had a dedicated safety person in the field. The other half, roughly, wore some form of organic vapor cartridge respirators, either split almost in half between full phase and half phase, and included both sprayers and helpers, and it was exclusively to the small contractors. We saw 2 helpers who didn't wear any respirators. In terms of dermal protection, almost all of them, with one exception, wore some sort of gloves. 3 mil thick nitrile gloves were the most common one, well, 82%. And latex was in 2 cases, and in one case, the helper again did not wear any gloves. They all wore coveralls and goggles. In terms of personal air sampling results, this table summarizes air concentrations, the distributions of measured concentrations for the isocyanate species of interest, phenyl isocyanate, the three MDI monomers, the MDI trimer, and the bottom row is a calculated total NCO based on those four measured analytes. It's another way of expressing their concentration. It's not a true total NCO in the sense that it does not include the many new species of isocyanates that are formed in this process. Sprayers had slightly higher exposures relative to helpers, although that wasn't statistically significantly different. And one of the highest species present in the aerosol was a 4,4' MDI with a geometric mean of about 20 microgram per cubic meter of air. If we look at the maxima in the range column, the highest value, personal value, was for phenyl isocyanates, 466 microgram per cubic meter of air, and then around 120 to 180 for other MDI species. So compared to existing exposure standards or guidelines, all samples were below the OSHA ceiling of 200 microgram per cubic meter of air for MDI. All samples, personal samples, were below the REL of 50 microgram per cubic meter of air. When adjusted to an 8-hour shift, assuming zero exposure for non-measured time, if we assumed the same measured concentration across the whole shift, then about 12% of samples would be above the NIOSH REL. Whereas if we look at the total isocyanate content for, again, I would like to emphasize, for those 5 measured species expressed as NCO, 33% of samples would be above the UK Health and Safety Executive reference of 20 microgram NCO per cubic meter, and 15% of them above the 70 microgram per cubic meter. The results of area sampling are summarized here, and one could see generally lower exposures for area samples with a remarkable variability, the geometric standard deviation in the order of 7 to 20. However, I would like to emphasize that the maxima for some of these species were high, actually higher than in all measured personal samples. For phenyl isocyanate, the maximum value was 1.5 milligram per cubic meter, and 240 for MDA, which indicates that bystanders and other crews that may happen to work in that area could get a decent amount of isocyanate exposure if they enter an active sort of a spray, SPF spray or application site, and so that that area should be restricted from access to non-essential personnel. The results of the glove dosimeter samples indicate potential for significant contamination of the hands with values that are in the order of milligrams of isocyanate species per pair of glove. We have normalized this over a 30-minute period because the amount of time that the glove was worn varied considerably depending on the scenario. However, the main message here is that as one would expect and would observe, there is a high potential for dermal exposure. With sprayers getting somewhat higher levels of loading of these isocyanate species on their gloves or hands relative to helpers. One of the more meaningful measures of of this effort is to look at the levels of urinary MDA pre and post shift, because that is a way to look at whether the workers are being exposed and whether the PPEs are working, or whether they're getting exposure through some other sort of means. The graph shows the distribution of urinary MDA values adjusted for creatinine, and when compared to the existing biomonitoring guidance values of about 1 micromole MDA per mole creatinine, about 26% of the urine samples were above the biomonitoring guidance value, and about 4% of them were above the value of 4 that is used in Germany. So the main message here is that these workers are still getting some exposure, and in about 1 in 4 of them, or 25%, these exposures are above what is considered good hygiene practices. We then looked further to see whether there was any trend related to the use of respirator types and the job or task. The graph to the right shows that there wasn't, and we looked at the difference post-shift versus pre-shift as in a log scale here. So if the difference is 0, then the average will be around, the distribution will be around a value of 0. Sprayers, actually workers who used supplied air respirator seemed to have smaller variability in their pre and post values of MDA compared to workers who wore OVC, but they were not, the distributions were not statistically significantly different. The graph to the left stratifies those that difference by task for bystanders, the helpers, the sprayers. We also have 2 individuals here who, during the study, they conducted only trimming and remediation of a closed cell foam. And here again, we see not a huge difference between the 3 groups, with sprayers having a little bit more than the helpers, and a little bit more variability as well. So we now are switching attention to a flame retardant, which is in part B, and I will present some results from the pilot study we did on the exposure levels and biomarker levels, urinary biomarker levels for one of the flame retardants, TCPP. At the time when this pilot study was done, we had access to 6 sites. They all belong to sort of large contractors, and every one of these individuals wore a supplied air respirator and was adhering to the best industrial practices. When we look at what types of flame retardants were used in these sites, TCPP was and remained the more frequently seen flame retardant, and that's the reason why we focused on TCPP. Its chemical structure is shown to the right here. There are also mentioning of some other types of flame retardants, but the concentration of TCPP also varied considerably depending on the cell type and possibly the formulator. TCPP has 2 known metabolites that are specific to TCPP. The one to the left, called BCPP, has a hydroxyl group taking place of one of these 1-chloropropyl groups, whereas the one to the right has a chlorine group on the chloropropyl substituted with an OH group. The one at the bottom is another flame retardant that is used in SPFs. we analyzed the same set of samples, personal samples, using mass spectrometry with multiple reaction monitoring, similar to what we did for isocyanates. The air and glove samples were analyzed in our lab, whereas the biomarkers, actually a panel of biomarkers for flame retardants, including the 2 in blue that belong to TCPP, as well as 3 other ones that belong to other types of flame retardants, were analyzed in Professor Stapleton's lab at Duke University. And so in terms of the sampling results, we see geometric means of about 300 micrograms of TCPP per cubic meter of air, and close to 19 milligrams of TCPP per pair of gloves. In terms of the urinary biomarkers, and this is an aggregate analysis, we have all pre and post are analyzed together. The 2 TCPP biomarkers have a geometric mean of 6.1 and 89, respectively, with GSDs of about 3 and 4, and wide ranges. The second one, with the long acronym, had a maximum of 700 nanogram per milliliter. I apologize, nanogram per milliliter. Of note here is that some other of biomarkers of flame retardants were elevated in this group of SPF applicators. And to give us an idea of how much higher they were, one can compare them to some typical geometric mean values of the distributions in the general population, as well as in the general indoor air. And the main message is that this cohort of SPF workers is getting a considerable amount of TCPP uptake. So the question, and again, the reminder here is that this group of workers was adhering to the best industry practices, supplied air respirators, gloves, coveralls, goggles. And so the question was, what is driving this uptake in this cohort of workers? We looked at both the difference between pre and post shift. The table summarizes the p-values for those comparisons, and the post shift values for both biomarkers of TCPP were significantly higher than the pre-shift values, and that remained true whether we adjusted for creatinine or for specific gravity. So this clearly indicated some active uptake of TCPP during the shift. And to further confirm this conclusion, none of the other biomarkers of flame retardants in these workers was elevated during the shift, although it was elevated in general. And the distributions were different. So in using linear mixed models, we tested whether the air or the dermal were driving this increase in the urinary levels of TCPP, and the amount of TCPP in gloves was strongly associated with the levels of post-shift BCPP, the biomarkers, remained true for both of them, indicating that this is likely driven by dermal absorption or even perhaps hand-to-mouth pathways, because dermal contamination is considerable. Now, because we did not measure the actual dermal exposures, a legitimate question is, what is the meaning of this potential dermal exposure? Well, for one, if we understand where exposure may occur, we have an opportunity to intervene. Secondly, we also need to understand whether the garments that are typically used in the SPF will provide sufficient protection. To the best of our knowledge, there are no permeation data that use realistic formulations of SPF, in this garment, and so we undertook a pilot study where we tested the permeation of a number of garments, including latex gloves and nitrile and vinyl, 3 mil thick, that are commonly available in Home Depot type of stores, and we've seen being used in the field, as well as 2 types of polypropylene coveralls, relying on design by Professor Yost at University of Washington, who was kind enough to help us with the permeation panel. And one of our doctoral students actually spent the whole summer setting up these cells, spraying them. Here's the cell before with a membrane before the foam has been applied, and then the 2-pack spray pack low pressure spray foam was applied, and then those impregnated filters underneath the membrane were collected at different time points, and then we analyzed them for both isocyanate markers, well, isocyanates, and for TCPP. The graphs here summarize the results of those, normalized as sort of slopes. And the graph on the left shows the 3 different types of gloves: latex, nitrile, and vinyl. And the main message here is clear, that the most protective of the 3 gloves was the nitrile glove, which showed the lowest rate of permeation, whereas the worst performing ones were the latex gloves with a permeation rate of about 4 microgram per square centimeter surface per minute. The 2 coveralls performed relatively similarly, with the polypropylene one being more protective than the Tyvek, but the differences were not huge, I think. When we looked at the behavior of different isocyanate types, we saw the phenyl isocyanate had the highest permeation rate of all, followed by 4,4' MDI. And that can be seen in the slope and as well as the absolute values. Overall, however, the permeation rates were low in the nanogram per square centimeter range, and the cumulative permeation over the test period up to 30 minutes was below the 1 microgram total NCO, which is, so the 1 microgram is the ASTM standard for acceptable sort of permeability. Similar results with TCPP, with both polypropylene and nitrile gloves showing permeability, with the nitrile glove being similar to what we saw for isocyanates, but at a higher permeation rate, and similar, and also polypropylene coverall permeation rates of about 1 nanogram per square centimeter per minute. So the main message. So the overall airborne personal exposure levels to sprayers and helpers were below the OSHA PL. All of them were below the OSHA PL. However, if we express that exposure as total NCO, even with the understanding that the total NCO we calculated here is a gross underestimation of the real NCO, total NCO, we still have quite a substantive percentage of samples being above the standard. There is high potential for dermal exposure to both isocyanates and TCPP. We found about a quarter of the workers we tested have urinary MDA levels that are considered above the value of 1 micromole per mole creatinine, and that is the value for sort of acceptable, good hygiene practices in place. So I guess one of the more important questions here is what else can be done better? Where are these exposures coming from? And at least my personal take on this is that most likely the inhalation exposure may be coming from intermittent exposures to overspray aerosol. It may be related to long-term dermal contribution, and even possibly from inhalation of resuspended dust. This we haven't tested, but there is circumstantial evidence to suggest that that may be somewhat at play here. We found no association between airborne and dermal exposure to MDI with a urinary MDA, which is contrary to what we found with TCPP levels, where dermal exposure seems to be driving the active uptake during the shift. And the nitrile gloves and the breathable coveralls do provide significant protection against sort of dermal exposure for both isocyanates and TCPP. We would like to promote the use of nitrile gloves that are thicker, the 5 mil ones, because they provide better resistance to wear and tear, whereas the 3 mil nitrile gloves, in a number of cases we observed, are torn apart fairly easily, creating opportunity for actual dermal contact with them. But even the worst of them, the vinyl, for example, gloves provided at least almost 100 times a protection factor when compared to the isocyanate loading on the one side of the membrane. We still think there are some opportunities for improving improving practices and reducing exposures. Training and awareness on best practices, especially for small contractors where there is some immigrant workforce. We've run into people who don't speak English, who come from another place, another culture where safety is at different standards, and limiting access to SPF sites for non-essential personnel is also a practice that we saw implemented properly in the case of large contractors with a health and safety specialist on site, but we didn't see it being implemented across the board. Reduction of dermal exposure, I believe, is an important way to hopefully cut down on some of potential uptake of isocyanates and associated effects that goes with that. Contaminated tools, not contacting the fresh foam with bare hands would be another one. And for understandable reasons, the focus historically has been on isocyanates. However, I think we have reached a point where we ought to look more carefully at other Part B components, and the amine catalyst and TCPP are high up on that list. I mentioned we have done some pilot work with remediation of closed cell foam using powered brush tools and saws. We know for a fact that those processes generate significant amounts of dust, nanoaerosols that are rather sticky and buoyant. We have looked at the isocyanate content of them, and the data we have suggests that they have quantifiable NCO groups of them. We believe it is worth to look closer into that scenario, especially from the standpoint of how to improve practices and to reduce inhalation exposure to such SPF-driven aerosols. They are loaded with SPF, with TCPP, for example. And as an analytical chemist who has spent a lot of time in the lab developing tools and developing methods and improving methods and adopting them for these challenging applications, I still feel that we need to invest more in developing both analytical tools and samplers and approaches that are suitable for these challenging applications. And among the list of those challenges is definitely developing a route-specific biomarker or a specific biomarker of exposure for isocyanates, which will help us untangle contribution of different exposure routes and the like. And with that, I would like to thank you for your attention and participation, as well as I would like to thank all of our collaborators, supporters, applicators, contractors, and our many collaborators who have helped us tremendously during this past couple of years to generate the data that we were able to share with you today. And with that, I'm turning it back to you, Jessica, and I would be happy to deal with or address some Q&A. Thank you.
Great. Thanks, Dhimiter. We have had a few questions rolling in during your presentation. So first thing I'm going to say is regarding whether or not the presentation will be available. We will not be sending out the presentation itself, since, as Dr. Bello mentioned, some of the data is still getting cleaned up, but we will be sending out a recording of the event as it happened today. So I'll be emailing that out by the end of the week to everyone who attended and registered. And then to the presentation-specific questions, I think the first one is actually referring to the charts on around slide 21, and it's just, I'm going to see if I can go back to slide.
Too fast, 21.
So can you just explain what the column GM and in parentheses GSD, what that stands for?
Let me see. Is it this one related to dermal?
On a couple of the slides, I believe.
Okay. Well, it's a fair question, and I'm aware that because of time pressures, I went through slides rather quickly, but I was advised to do that. So the GM stands for geometric mean, and it's a measure. That's the question, correct? What is GM and GSD? So the GM stands for geometric mean, is a measure of the central tendency of the distribution in of a skewed distribution. So in lay terms, is a kind of average for a right tail distribution. And the GSD is a measure of how variable the data or varied the data is. So how huge the difference between, how long the tail is, in other words. So a GSD of 3 is I guess, okay for personal exposures in workplaces, a little bit on the high end, but sort of small in general. But a GSD of 20, as we saw in some cases, it's remarkably high and indicates that there will always be some cases where one would run into very high exposure levels.
Okay. Thank you. I'm seeing a couple of questions that are related less to the presentation that I just want to touch on real quick. So one of them is, would we repeat the webinar? And actually, Dr. Bello and I have talked about that. And if there is enough interest, I think that is something that we would be open to. So if you know people that were unable to attend, or if you are interested in a repeat, please just email me about that, and we will figure out whether that's something we can make happen. And then the other one is related to the future webinars I mentioned. It says, at some point in time, will you have another webinar on isocyanate and epoxy coatings? So, Dr. Bello, I don't know if you want to briefly just talk about the additional webinars that we had discussed possibly doing in the future. Would they touch on epoxy coatings or not?
So the next in line, which will be perhaps sometime next year, will be on industrial coatings using isocyanates. such as bridges and reactor domes and tanks and stuff. We are still actively sampling those scenarios. I know there are practically no exposure data there, and some of the things we see and we find are rather intriguing. So I would like to use this opportunity to reach out to the audience for if you are involved in some of these activities, if you are interested in participating, or if you would like to help us, or if you know someone who is involved, we are very interested in sampling more of these scenarios. So that's the second planned webinar, hopefully in a year's time, because we are still analyzing samples and the data, and we would like to get some more. We have started work with epoxies, and some of the applications we are looking at right now are related to coatings, as well as both steel structures and floor coatings and glues and mortars. That will be perhaps the last one. It won't happen anytime soon, I guess. Perhaps we're looking a year and a half to 2 years for that, since we are early on in that process.
Okay. The next question is, do you have results specific to roofers from this part of the project?
So we were particularly interested in monitoring roofing activities. It turns out roofing is not commonly done in the New England area. So roofing is done, as from what we've been told, in the warmer parts of the country. So we haven't been able to find a true roofing application. The closest we have gotten is training on sort of a roofing application, which is sort of a simulation. And there we have some data. And from, I don't remember the results. I'm sure we will include those in the publications, but the exposure levels there may be similar to the range of concentrations we see with the regular insulation foam. But we don't have many data points on that, unfortunately.
Okay, thank you. The next question is, can you relate the permeation study results to what you found inside the glove/the dosimeter study?
So I would like to clarify again that the design was to look at what may end up on top of the glove, and that's what the dosimeter does. It collects the amount of isocyanate that would normally end up on the hands, on the gloves of a worker. And if the worker doesn't wear any gloves, then the dosimeter would measure the amount that ends up in the hands. We did not measure the amount of isocyanates underneath the gloves, and the reason was, there were two reasons, but one of them was that given how demanding our study design is, it was logistically very difficult to collect the data. And so by knowing typically how much isocyanate ends up on the outside of the glove, and by studying the permeation rates, we have some idea as to whether permeation of isocyanates in intact gloves occurs or not.
Okay. Related to the earlier questions about roofing and epoxies, someone has asked, there are new roofing systems that use epoxies. Do you have any plans to study those?
Yes, we are quite open to studying the questions that are relevant to those who apply them. And if the person who asked the question is still online, we would encourage you to help us to study those questions. This is the best time to do that, and we remain very committed to doing the best possible science that we can, and to provide answers that really help you guys do your job safer. As you may have realized by now, based on the presentation on isocyanates, we are driven in our recommendations by the actual data, and we are open and transparent, and our motivation is to help construction crews do their job safer.
Okay, thank you. The next question is, when we observe workers spraying, many of the instances they are in a confined area. What types of testing was done in a confined space? Or was any in a confined space?
Exactly. I'm not sure what the intention of the question is, but I'll try to answer it to the best I can.
I think he's asking, like, how true are the study circumstances to real life?
Well, they are the real life. They are the real life. We had, and one of the pictures depicts that, but we had half of the study participants were crawling underneath the first floor of a one-floor house or in tight spaces where they could even stand up at all. And they were rotating 360 degrees on their belly and their back and belly and back with the gun right at their nose. And in those cases, we have actual personal exposure data, the monitor on their chest, collecting data, glove samples. What I forgot to mention, I guess, is we also collected sort of patches in for a number of coveralls in 10 different anatomical parts to give us an idea of whether isocyanate and foam was deposited in other parts, like the back and the legs and the arms and the like. So these data are the real thing. And we normally send those results to the contractors and the workers we sample, and we follow up with them. But this is real, and it covers the whole spectrum of conditions and foam types and types of formulations, I guess. So this is real, is the bottom line.
Okay. Do you have an idea around when these results will be published?
Well, we have 2 manuscripts that will be submitted fairly soon, and the third one with the actual MDI and spray forms that is in preparation. The fact that we have most of the data analyzed and summarized will make it easier for us now to submit, but our interest is to get the data out as soon as possible.
Great. Do you have time, Dr. Bello, to keep going through questions? We still have several more, but I know that we're approaching an hour.
I'm fine. I'm fine.
Okay. Okay. Next question is, do you think strict personal hygiene measures such as showering after application would reduce overall dermal exposures and therefore reduce systemic absorption of the ISD and TCPP?
Well, this is a personal, professional judgment now, because we don't have data and we'd prefer to have data. But I do think that if the contaminant is still on the outer layer of the skin, then showering will definitely reduce that. However, I don't think showering will be able to remove the fraction of contaminant that has penetrated deeper into the skin. That I don't know how will play out, but I think showering at the end of the spray period is something that would only reduce exposures, I guess. My hesitancy here is that showering with hot water may do other things to skin permeability, but it will certainly remove dust, and I think it will remove the TCPP on the skin as well, and it will destroy isocyanate groups. So my guess is it most likely will help.
Okay, great. Are there any specific kinds of tasks that you notice contribute more to dermal exposure? So tasks where they might have contact with raw product or where they might remove their gloves and have dermal exposure?
So I think an easy answer to that in regards to TCPP is, from what I've seen, so because the foam, the cured foam itself is seen as non-hazardous, the crews are sort of accustomed to touching it with bare hands. And because there is so much TCPP in the foam, I suspect that that practice may contribute to dermal uptake of TCPP. So I would suggest perhaps refraining from touching the freshly cured foam on site with bare hands. I also think that another sort of task where there may be contact with the raw material is the cleaning of the gun, because they take it apart and then they try to sort of unblock it, and handling of the drums. Actually, in some other scenarios, we have found handling of empty drums to be a major source of dermal contamination, because there you have raw material and direct contact with the pure part A. And the last one that we see it often, although it in our experience has been done with sort of gloved hands, is punching through the freshly sprayed foam to probe for air pockets, and either with a gun or with a hand to see if the foam is doesn't have air pockets. And we suspect that may deliver some isocyanates in the wrist elbow area, which is between the glove and the coverall. At least in some small contractors, we suspect that to be the case.
Okay. Next question is, did you ask any workers or happen to make observations about whether they experienced contact dermatitis or asthma?
The study is not focusing on health effects, so we don't systematically investigate that now. However, in a number of occasions, the workers themselves approach us and were sharing with us their concerns related to dermal exposure and contact dermatitis. And so I have seen for a fact that a number of these workers have contact dermatitis. It is difficult to tell from the appearance whether it's irritant or allergic, but I would suspect that at least in one of the several cases I have seen, it may be allergic in nature, and I judge this based on the delay in the appearance of symptoms, the type of symptoms, and the like. But there is contact dermatitis in some of these workers. And we actually, in one case, we worked with one of them to try and reduce dermal exposure to, and he was getting that on his right above the wrist.
Okay. What type of air purifying respirator cartridges were utilized for isocyanate protection during foam spraying operations?
Could you repeat it again, the first part of it?
What type of air purifying respirator cartridges were utilized?
Well, so in all our sites, there was only one that adopted, towards the end of the study, powered air purifying respirator, because we encouraged them to consider that over an organic vapor cartridge. So it was either a supplied air respirator, where the purified air was supplied through a hose. That was the only supplied air purifying system that we saw. And the rest was organic vapor cartridge respirators, like the ones you buy for $30 at Home Depot, where you have two organic charcoal cartridges attached to the respirator.
Okay, this actually relates to another question, which is the airborne concentrations you measured were not terribly alarming, but the health effects from sensitization are quite serious. Do you have any recommendations on appropriate respirators?
This is a tough question to answer because one of the questions we've struggled with or have been grappling for some time is, well, what kind of additional engineering controls could we apply to further reduce inhalation exposures? And NIOSH recommends supplied air respirators, and half of our crew was using supplied air respirators, was using the best industry practices, essentially. The issue with supplied air respirators is that, well, they have long hoses, and it's more expensive to utilize them. And roughly, based on our survey, half of the crews, almost all small contractors, were relying on organic vapor cartridge respirators. Now, the data also showed no huge differences in the biomonitoring results between those who wore supplied air and those who didn't wear them. So if we are strictly basing our recommendation on the data we have, it seems that even the OVC respirators are doing a decent job. So perhaps a level up from the OVC respirators may be to use a powered air purifying respirator, which is a hybrid between the supplied air respirators and the OVC. And that has the advantage that it theoretically may provide a better protection factor than OVC, and it's easier and it's a lot more portable. However, there are no actual data to sort of firm our recommendation on that. So I understand this is a fairly legitimate question, and it's an important one with serious practical implications. The reality is that on the ground, many crews simply cannot comply with the use of supplied air respirators that rely on outside compressors and filtered air and long lines, and so they go to the next option. Because that is a practical limitation, my personal opinion or thinking at this moment is that it is perhaps logical to consider promoting an upgrade from organic vapor cartridge respirators to a powered air purifying respirator. I hope this answers part of the question, but
that last question was from Bruce Lippy here at CPWR, so I'm sure that he will be following up with you in the future as well. Okay, we have a couple more. Another recommendation question. Do you have any recommendations on barrier cream?
This is an interesting question. We haven't done any active research on barrier creams recently, but about a decade ago, we were looking at them in the context of prevention of dermal exposure in auto body shops. I do not feel comfortable answering that question because I don't think there is, at least I have not seen any solid data on the efficacy of barrier creams. It could be a barrier cream, but on the other hand, there is always the potential that the barrier cream itself may serve as a reservoir for some of these ingredients. Let's say if that a few micron layer of cream around the hand gets loaded with TCPP, then that could serve as a reservoir in the skin. So for delivering some of these to the skin. I would rather want to see data or generate data on the efficacy of barrier creams. And I'm sorry I cannot, I don't have an answer to that.
Okay, no problem, thank you. Let's see. Plumbers and pipe fitters sometimes use a heat source that heats up the cured SPF. Is there any exposure information for this activity?
So yes, they heat both the drums as well as the lines. And we have collected a few area samples inside the truck with the intention that just to verify whether there are any MDI vapors generated in that process. If I recall correctly, those were among the very few samples that were non-detectable in our case. Heating of the isocyanate may, and the exothermic reaction at the point of application may explain the unexpectedly high air levels for phenyl isocyanate, which is technically an impurity in the polymeric MDI foam, but for which we measured some surprisingly high levels. I think it's evaporation of those impurities because of the heating and the exothermic reaction. Apart from that, I think I don't see any other reason for concern there, as long as the systems are operating closed as they are supposed to.
Okay, thank you. So I think that was actually the end of our questions. We did have a comment that I want to refer people to at the bottom of the Q&A, which you can all see on your screens, referencing a NIOSH PowerPoint with regard to what you were saying, Dr. Bello, about the use of supplied air for indoor applications. So the PowerPoint includes some information about NIOSH's official position, which sort of just reiterates what you were talking about. And I also wanted to thank everyone. There have been a bunch of comments coming in about what a great presentation this has been. And again, I would also like to thank you, Dr. Bello. It really has been a very informative presentation, and I appreciate you sticking it out to answer all the questions. We also have at least one person who is willing to help with some future studies. So I will be following up with you, or Dhimiter or one of us will be. And any other questions that I may have missed, I'll look through and we will certainly follow up on anything that we didn't cover. If you have any questions that were not asked, feel free to email me and I will get in touch with Dr. Bello and we will get you the best answers possible. So thanks again, Dr. Bello, and thank you all for joining.
Thank you, and have a great day.